Okay, so we are about to start block number 5. These are 4 Mission 7 experiments.
Next up is oral presentation number 21 from Austin, Texas. It's a Mission 7 flight experiment with 8th grade student researchers.
Hello, everyone. My name is Steven Fonserrata.
I'm Kyle.
I'm Miles.
And I'm Will.
And we'd like to thank you for being here today while we talk about our presentation, the growth and development of the heterotrophic algae Neochloris oleoabundans in microgravity. In order to have an experiment, we must first have a couple of questions, and the questions that we were hoping to answer for this project were, will microgravity change the way our algae grows? Will microgravity change the lipid content of the algae? And could algae be a potential source of fuel or food on long-duration space flights? Kyle, if you'd like to talk about the implications of this.
So the reason we chose to investigate algae is because it's a very versatile resource. It's very rare to find an organism that can provide nutrition, fuel, oxygen, and medicine. On top of that, algae grows very quickly and it's relatively easy to culture. In the future, algae is expected to replace fossil fuels and fight diseases we may not have answers to today. One major problem for the future is how space travel will work. How will we have the resources to travel to Mars? What's needed to create a self-sustaining space colony? Algae could potentially solve these questions and more, but before we can use algae in space travel, we have to test it in microgravity.
Our experiment consists of 3 main ingredients. Our first and most important ingredient is our algae, Neochloris oleoabundans, which we'll talk a bit more about later. Next, if we wanted our algae to grow, we would need to have a food source for this. We decided to use the universal energy source, glucose. Finally, if we wanted something reliable to measure down here on Earth, we would need to fix the results on the way back down from the ISS. And to do this, we decided to use the growth inhibitor puromycin solution. Now Will talk a bit more about our algae.
Okay, so we chose our algae because it's a very versatile algae. It can be grown both with and without light, particularly without light, because there isn't light in the ISS, or rather the area where the experiments will be performed, so it needs to be able to grow without it. And also, it can be grown in confined spaces, such as the FMEs that we will be conducting our experiments in. And finally, the algae has a variety of uses, such as fuel and energy, and perhaps even food.
Procedures.
Oh yes. Now the procedures are fairly straightforward. They'll first unclamp the first clamp to release the glucose into the algae so it has something to produce energy with. It'll grow for 9 days, and then after those 9 days, they will unclamp the second clamp and releasing the puromycin, which will inhibit the growth so that they can send it back down to Earth where we can analyze the results.
As for analysis, we will be taking 4 1 milliliter samples of our algae, Neochloris oleoabundans, and putting 2 in pre-weighed aluminum dishes to be baked and dehydrated so that we can find the dry cell weight of the algae. The other 2 will be put in tubes and exposed to chloroform and methanol as detailed in the Fulch extraction procedure, and so that we can extract the lipids, and the results will be compared with our ground truth experiment here on Earth.
Our experiment setup and analysis is all done with the help of Dr. Rika Connolly and the Center for Electromechanics, or CHEM. CHEM investigates the growth and uses of algae. Dr. Connolly owns a research company called GrowFizz that studies the commercial implications of algae. And now we'll talk about what we learned.
Being a part of the SSEP mission has provided us with a new understanding of experiment design and what a successful experiment takes to develop. I think we learned a lot about authentic, real, the kind of things that people do in a lab for science that we don't really get to do in school. And personally, I thought that it was a lot of fun thinking through the procedures and brainstorming with my team.
As Steven said, it was a lot of fun working in the lab. We got to learn a lot about the various equipment like centrifuges and high-powered microscopes. It was really something that we've never done anything like before.
Of course, we also learned about the ISS and microgravity.
And finally, we learned many things about the way algae grows and its various commercial applications, such as fuel, energy, and food, again.
Finally, we would like to thank all of our partners. You can look up on either of the PowerPoints and see them.
And now we'll do some Q&A. So if you have any questions, come up to the microphone.
Mike Buchanan from Austin, Texas. What was the price of this experiment?
It was somewhere around $450 to $500. As far as science on the International Space Station goes, I'd like to think that's pretty cheap.
Thank you.
My name is Steph Tivnan, and this is Sam Bracton, and we're from Massachusetts. We were wondering what test you're running to be able to tell, what was it, the dry cell weight?
We will be weighing the aluminum dishes after we've baked the algae inside so that we can compare the weight of the aluminum dish before and after the algae is introduced, and then we will subtract the weight of the dish previously from the weight of the dish with the algae so that we can get the weight of the algae that we have.
Thank you.
Thank you.